Welcome to Face Forward, the podcast, the series that takes you behind the scenes of the riveting world of clinical trial management. I am your host, Valerie Coveney, and I am thrilled to be your guide on this journey through the nuances of planning, executing, and navigating the clinical research landscape.
So welcome to today's episode as we explore the future of skin diagnostic through the emerging science of biomarker sampling. Our guests are Simon Grant, CEO of Ascilion, and Théo Boyer, Senior Project Coordinator at Indero. With backgrounds in device development and clinical research, they offer valuable insights into how sampling dermal ISF could open new doors in dermatology. So welcome to you both.
It's Simon here. Thanks for having me on.
Thank you very much. Glad to be here.
Amazing. So let's get started here with our first question. So Simon, what led you to start working at Ascilion?
Well, I've been working in medical devices for many years, and I when I started to get involved with Ascilion, I saw that there was really this untapped potential from within dermal interstitial fluid, or ISF, dermal ISF as we call it. ISF contains almost all of the biomarkers in blood, but it's difficult to access, and I think, and so what the company I work for, Ascilion, does is it's best in the world at extracting that fluid. So what we call sample the fluid, and we do that in a way that is non-traumatic for the skin and can be repeated. And what that enables us to do is to get a snapshot of what's happening in the body at just that time. We can measure thousands of potentially different biomarkers, not just within skin disease, but within cardiovascular, within hormones, within all different kinds of things. And I think that's really the potential and the beauty of ISF, is that it reflects almost all the biomarkers that you see in blood are reflected in ISF. And that's what, when I looked at this company and what it was working with, that's what I saw as really an untapped potential that is going to become really important as we go forward, and people want to see more about what's happening in their body, not just what we call physiologic biomarkers like activity or temperature or whatever, but also molecular biomarkers like glucose is done today, but also could be hormones, can be cardiovascular biomarkers, and so on. So that's the thing that Ascilion saw, that this is coming, and it's going to be ubiquitous. I think it's going to be everywhere, but it's going to take time, obviously. That's the potential, I think, and the interesting thing about ISF. It gives you this view into the body, and we can access it or sample it in a really, minimally invasive way.
Mm-hmm. Yes, and Théo, how does your background in molecular medicine and project coordination support this innovation?
So I have around 10 years of academic background in biochemistry and molecular medicine. I've always been interested in improving people's lives through healthcare. And I believe that the future innovation will really contribute to this. So that's why I'm very excited with the Ascilion technology. During the last year, I've been lucky to work on multiple early phase trials and develop some knowledge on all the challenges related to this. So what I love with early phase studies is that they are really showing innovation, bringing innovation to the clinical research world, and also it's every study is unique and has its own challenges. So I really believe that my academic background will help me to understand what's the objective of the study, and also my experience with CRO at Indero helps me to counter the challenges through project management.
Okay, and how did both your paths cross, and how does your collaboration look like? What does it look like?
Yeah, I guess we met through a customer. A joint customer. So we had a requirement. We had interest from a client in being able to measure what's happening in the skin without having to do biopsies or tape stripping, and it's a relatively new method. So we wanted to kind of work on a project together to show the value of this method, and that's how we met, I guess, from my side at least.
Yeah, I share this. So my director proposed this project. She talked about the innovation, and I was really interested to it. So she assigned me on that project. I've been lucky to meet Simon in May at the SID, so it was like also great to meet you in person and being able to discuss like the technology, because we have already been started to work on the project. So this is how the collaboration is going so far. So Simon, can you please explain what's the dermal interstitial fluid?
Yeah, it's a good question. Interstitial fluid is a bridge between the circulatory system and cells. So all the nutrients that get delivered to cells pass through this interstitial fluid, and they don't come directly from blood. And that is not often understood, that there's this interstitial fluid, which is the kind of the bridge between the circulatory system and the cells. So we know that what is in the circulatory system is expressed in this interstitial fluid. So we can see almost all of the biomarkers that exist that you can measure in blood, you can measure in this interstitial fluid, because it carries everything back and forth to the cells and then back to the circulatory system. So this fluid is actually more than double the amount of interstitial fluid in the body than blood. It's very hard to access, and that's been our challenge.
Thank you very much for the explanation. And I wanted to know how was born like the focus on the d-ISF or the application on dermatology?
Yeah, I think it originally came from the same realization that there was a kind of an untapped way to access the biomarkers in the body. And I think what you can use is to illustrate this is continuous glucose monitoring, or CGM. CGM measures in interstitial fluid. It doesn't measure in blood. It's very difficult to measure continuously in blood in the body. So we use interstitial fluid because it reflects what's in blood, but it doesn't have the same problems as measuring in blood. So this is the first, I guess, application that really utilizes interstitial fluid, and you see how popular continuous glucose monitoring is. That measures glucose, but there are thousands of biomarkers in blood and also in interstitial fluid. And so that's the potential of interstitial fluid. It provides access to all of these biomarkers, nearly all the biomarkers that you see in blood, you see in interstitial fluid. So it gives us a different way, a really minimally invasive way to access those biomarkers.
That's very interesting. So if I understand, you can access all like the biomarkers within the d-ISF. The current gold standards to evaluate those biomarkers are usually like the biopsies or the tape strip sampling. What are the advantages of using this new technology to evaluate those biomarkers?
Yeah, we use a microneedle array, very, very small microneedles, and they penetrate just through the epidermis and into the upper dermis. They penetrate about 1/3 of a millimeter into the skin, so they're very minimally invasive, and so they don't cause pain, and you don't get trauma to the skin anywhere near what you would get with biopsies, for example, or skin tape stripping. And these are some of the alternatives that are used. So we provide a way to access the information that's contained in ISF without creating a lot of skin trauma. And I think that is what is of interest to most of our customers, is how can we access these biomarkers without creating a lot of trauma for the skin, and opening up this new set of information that is available from ISF.
So it really improved like subject comfort on those samplings.
Yeah, it's really well tolerated by subjects, and I think one of the challenges, of course, with biopsies is that you can't do lots of biopsies. You might be able to do one, but you're not going to be able to do multiple biopsies. You can do sampling of ISF. What we call sampling is taking up ISF from the skin multiple times, and it's well tolerated by by patients and subjects. So this is one of the key advantages, is that you can do it without causing a lot of trauma, without pain. It's well tolerated, and I think it's accepted by clinicians as being much less invasive than the other methods that are currently available. The ISF itself has other advantages in terms of you're able to see local changes, and that's one of the big advantages of this. You can see local changes in the skin, but I think that is in combination with these advantages of being able to sample multiple times and with good patient compliance.
That's exciting. This is really promising for the future. I was wondering if you identify current challenges that you're working on to improve this technology in the future.
Yeah, I mean, the technology today, I think we are working really hard to make it easier to use. I think that's the we want it to be quicker. It currently takes about 15 to 20 minutes to take a sample, but then we get enough of a sample that you can usually go and analyze. We look to try to increase the volume of the sample of the ISF that we extract. So we extract ISF through from the skin, and we try all the time to increase the volume so we can deliver, that we can do it as quickly and easily as possible. And I think those are the key focus areas for us. And then on the other side, there is how to use this information. It's a new type of information that obviously requires some validation, and it needs to be accepted by scientifically and so on. So we work with a lot of different groups. We have 18 different trials running at the moment, for example, and all of them are looking at slightly different things. But they're all adding to this body of evidence about what's in ISF and what it can help us explain in the body. And just one other thing, and then down the road, we see that what glucose monitoring shows us, the CGM, is that the value of being able to monitor a molecular biomarker, multiple times or continuously or regularly, is extremely valuable, obviously for diabetic patients. But what we see is we get the question all the time, CGM is for glucose. Why can't I do that with cortisol or progesterone or whatever? That people more and more understand that the ability to measure molecular biomarkers as well as the normal biomarkers that you might measure with your Apple Watch or whatever is an extremely interesting proposition. And we see more and more that we believe that there will be a move towards measuring many different biomarkers, both the biomarkers that you traditionally get with wearables, but also molecular biomarkers going forward. And it's a combination of these two that really has power and has value to understand what's going on in the body. So we can go from having testing being done in the lab or in the hospital to more something that is done regularly, perhaps at the doctor's office, point of care, or even in the home. And that's the journey that we believe. That's what we believe is going to happen over time. That there will be more and more biomarkers, molecular biomarkers, monitored, measured, and monitored at the same time as other biomarkers, as other wearable biomarkers. So it's going to converge over time, and we see all the time. We see signs that this process is ongoing.
Cool. So there is a lot to come.
Yeah, and that's a little bit aside from the clinical work, but this is. I think it's important to set the vision for why we believe what we see happening in the future, and our role in that future is to support that and to make sure that the data is there and the tools are there to exploit this opportunity.
Okay. So thank you very much, Simon, for all those details providing on the technology and the application of the ISF and what's coming for the future. Currently, I was wondering what kind of dermatology conditions are you targeting with the d-ISF sampling?
Yeah, I think that if you look at all our customers, I mean, we have customers from a lot of different areas, from hormones, from cardiovascular, but at least a third of our partners, our customers, are working with inflammatory skin disease in one way or another. And there you have atopic dermatitis, of course, but even prurigo nodularis, psoriasis, seborrheic dermatitis. So there's lots of different conditions. And I think that there are two factors here that are important. There is, sometimes you want to quantify biomarkers in skin, in healthy skin, but then other times you want to compare that and you want to look at the progression or the changes that occur in lesions, atopic dermatitis, eczema lesions, for example. You want to measure what's actually happening in that lesion. And I think that though that's harder to do, it's still possible. And we have several studies happening within this space right now. And it's about atopic dermatitis, but it's also about psoriasis. It's about these other inflammatory skin diseases. And this opens up a completely new kind of possibility for clinicians because they can non-traumatically investigate what's happening. They don't have to take a biopsy. They can sample this interstitial fluid from the lesion or around the lesion or in healthy parts of the body, and they can compare and contrast and see what's the difference between what we see in a lesion or an AD sufferer compared to a healthy patient or healthy skin. So I hope that explains it, but really it gives you a different way to view what's happening with these conditions. And I think there's some recent data which has shown that ISF is a really good way. It's a really good way to investigate what's happening in the skin, and especially when it comes to bioavailability and so on. This is a real advantage, the ability to be able to sample, for example, an oral drug. You have an oral drug that works on the skin. We can actually sample, and we have done this, we can sample during a pharmacokinetic study and compare that to what you see in blood. And this is really exciting for customers and for pharmaceutical companies because they want to be able to see how much of my drug is actually making it to the upper dermis, because that's where it's supposed to be acting. And this is, we believe, so for systemic drugs, this is a really interesting additional opportunity to be able to measure what's happening in the skin, to say, okay, this much of my drug is actually making it to the dermis, and this is what is happening within, how it's affecting the biomarkers in the dermis. So everything is in ISF, and that's one of the beauties of it, is that it doesn't matter what you're looking for. If you're looking for hormones, you're looking for bioavailability of a drug, or you're looking for specific proteins or peptides or whatever, everything is contained in the drug. There are thousands of biomarkers in ISF, and that's one of the beauties, I think, of ISF as a medium.
Yeah, that's great. That's great to know that you can correct like a wide range of conditions and focus on like really specific biomarkers for each of them. And for the PK analysis, this is actually very interesting, and it will be very
it's new,
useful.
and it gives customers a new possibility. It's in a non-destructive, non-traumatic way for the skin, and you might sample 12 times during a PK study from the same patient in more or less the same place, and you can't do that with biopsy. You're not going to be able to tape strip 12 times or be able to biopsy 12 times, but you can do it with ISF sampling.
That's great. I was wondering if you identify like specific challenges with specific conditions such as like AD, alopecia, psoriasis, depending on the skin.
So there are a couple of kind of restrictions when it comes to the it's you have to be able to access obviously the area you want to sample from, and it should not it should be relatively flat. Now, that means that if you wanted, for example, to sample one finger or the back of the hand, which is very bony and uneven, that's a challenge for us. We are able to sample, pretty much anywhere on the body with the scalp. You have to shave a small area, but we can sample from the scalp, which might be interesting for alopecia areata, for example. We can sample from the arms, the legs. There's not a lot of restrictions on where you can sample, except for palms and soles of the feet. The skin is too thick there to sample. But for the most part, you can sample anywhere. the challenges become in very uneven areas, bony areas, for example. We need a relatively flat space of a couple of centimeters squared, and then we're usually able to sample. It's interesting, but we get better volumes, better sampling from older patients, and there's a couple of reasons for that. But that we still see a lot of variation between subjects in terms of the volume that we get. And our challenge, that has been one of our challenges, but we more all the time we're increasing the average amount that we get from a sampling, and what we get on average today is 15 microliters in 15 minutes, and that doesn't sound like much, but it's enough to do many analyses, and it's much more than you could sample, for example, a year or two ago. So we're really, leading the field in this, in terms of being able to reliably get reasonable amounts, enough sample volume to be able to do the analysis you want to do. And that's what you have to look at when you're working with sampling researchers. You need to say, okay, which analysis methods are we going to use? What does that mean in terms of volume requirements? Many of the proteomics studies, for example, if you're looking at proteins, they require only a few microliters. So that's a really good way, a good fit for us. And then some mass spec, it really depends. So it depends on the methods that you're using. And we help you through that whole process to understand what you need to do to get the volumes that you need to extract. So that's a part of what we do, is helping our customers to do their studies, to structure their studies in the right way so that they're going to get good results.
That's great to know that like multiple indication can benefit from like the technology and like all subject, because you said like we can sample multiple areas. And do you think like the d-ISF sample in analysis are like really sensitive in terms of biomarkers, targets, or identification?
Yeah, I think it depends on the biomarker, but so far we've been really happy with the results. We generally we look at about, or our customers, I should say, look at about 100 different things when they take an ISF sample and they run maybe an O-link panel of 90 proteins, and then they want to look at another marker. It might be, we for example, we've run 2 cardiovascular trials, and there you're looking at systemic cardiovascular biomarkers in skin. And these have been really successful. So we see NT-proBNP, for example. We can see it. We can see all of the cardiovascular proteins in the skin, and we can see how they change with exercise. And I think that's an indication of the potential value of ISF, is that it's giving you kind of a relatively real-time view of what's happening in the body without having to go in and take blood each time. You can do that through the skin, and you can do it with relatively little trauma to the skin, what we call microinvasively. Remember, we only penetrate 1/3 of the millimeter into the skin. So we do you have to go through the epidermis, but you're able to really access the ISF by only going, 1/3 of a millimeter into the skin.
And that's great that this technology can be applied for non-skin indications as well. Like the application is pretty wide.
Exactly. So cardiovascular, I gave as an example. We've had two studies there, really interesting studies. We have hormone studies, cortisol, for example. We have a large study happening. They're one of the largest studies looking at cortisol levels, and everything is happening from the skin. And we know that most biomarkers are represented, are detectable in ISF. And the work that is being done now is to figure out, okay, what are the levels in the skin, and how does that relate to the levels in blood or systemic levels? And that's where we're going to see a lot of activity in the coming years, is, okay, how do we link what we see in ISF to what's happening in the body, so that we can make that available to patients, and even to wellness, people who are interested in wellness, in real time, or be able to measure it without having to take blood, without having to send it away to a lab or go to a lab. So that looking down the road, that's where we believe this is going to go. Right now, our focus is clinical trials and working with partners to develop the knowledge base within ISF. But down the road, we believe that ISF is going to be a fundamental kind of cornerstone for work within wearables, within molecular diagnostics, molecular biomarker monitoring. Okay, so Théo, I have a question for you. What do you know? How does Indero support these applications? I mean, what do you see as the value for Indero and for your customers with this technology?
As you mentioned, like the d-ISF application is pretty wide. So I really see the value added in our studies, like mostly to replace some gold standards analysis method, sampling methods such as like the tape strips or biopsy, which we see in the clinical trials. Biopsy could be a challenge to recruit some subjects because they don't want to have like some skin sampled, or there is a high risk of infections. So with this technology, as this is minimally invasive, I really think that it could help boosting recruitment in our studies. And
yeah,
because
what we see, if I just jump in here, what we see from the customers we talk to is that it's getting harder and harder to get ethical approval to do multiple, especially multiple biopsies, and even in some cases biopsies. And I know that FDA, for example, released guidance this year for, which really kind of indicates that they're going to try and restrict biopsies going forward, unless they're absolutely necessary. Yeah.
Yeah, you have to justify the use of biopsy. So I really think this could be like a more friendly method for regulatory approval or ethical approval, as this is minimally invasive. And another application that could be good for clients is like the application of the d-ISF biomarkers and the accessibility of them, because as Simon mentioned before, there is some biomarkers that are like inaccessible through blood and that are available in the ISF, so that could like improve like the application of the samples. And I really think like the innovation bringing with this technology could like really add some values to clinical trials. I have one last question for you, Simon.
Yeah.
What would be the future for Ascilion and the PALSA system in the next years?
Yeah, we see PALSA as being the system we have today. Today what we do is we have a system which is used to extract ISF so that it can be analyzed. But what we see going down in the future is that there'll be more and more companies. First of all, researchers in the beginning, but eventually there'll be more and more customers wanting to be able to microinvasively kind of measure what's happening in the body without having to take blood. Okay, that's the first thing that we see is going to happen. And if these biomarkers are reflected in ISF, this is an obvious application for the technology. But down the road, I think that this thing with CGM, being able to monitor glucose, regularly, almost continuously, has really opened up a lot of, possibilities within metabolic health. And I think that there is going to be a drive to add more and more different biomarkers that can be measured. And you can do that in two ways. You can put lots of different sensors into the body, or you can extract ISF and everything is there. And we believe that is going to become more and more interesting for potential partners if we can find a way to utilize ISF to access all of these thousands of biomarkers that exist in the body. So, and that's our key. That's what we do. That's, we're really good at extracting this ISF so that you can then go and use it for whatever you want to use it for. And we're, application agnostic, as we say. You can use it for whatever you want. But we see there's going to, people are going to want to see more and more what's happening in their body, molecular biomarkers, not just how many steps I take, but what's happening in my body today. And there are lots of different applications within that space. So we believe that we will play an essential role in the development of that space.
Very interesting.
Amazing. So this concludes our episode for today. It was a pleasure having you both on the podcast. Thank you so much for the insightful conversation, and hopefully we get to see you again, Simon.
Yeah, great. It's been a pleasure, and we look forward to continuing to work together.
Thank you very much for your time, Simon, and it's a real pleasure to work on this successful study.
As we conclude another illuminating episode of Face Forward, we find ourselves at the crossroads of science and progress. Remember that behind the jargon and statistics lie stories of unwavering commitment, meticulous observation, and the pursuit of evidence that shapes our understanding of health and disease. Stay at the forefront of knowledge and innovation and follow Face Forward on your preferred platform. My name is Valerie Kovney. Thank you for joining us. Until next time.